Okay, our next speaker is Aydan Bailey, and he's going to be telling us, doing a deep dive into mouse brain data. Maybe we'll learn what mice are thinking about. It's like me, it's probably cheese, but maybe mice are deeper than I am. Go ahead.
Thank you. Okay, so my name is Aydan Bailey, and I'm not an expert in the things that we are doing at the lab, and I'm also not an expert with CHTC. So this is kind of my experience just getting familiar with CHTC, and I'll try and give you a brief overview of what it is that we're doing to the best of my ability. But first, just a little bit of introduction on me. I'm a second year CS PhD student. I'm working with the Wisconsin Institute for Sleep and Consciousness, or the Tononi Lab. And initially, we at the lab were interested in using CHTC for PyPhi, which is the in-house software that we use to run calculations for their theory of consciousness, which is very interesting. That was the reason that I was interested in working with the lab in the first place. But yeah, it's very computationally intensive. And so we actually initially started with HPC for that, and then discovered, hey, this is actually pretty useful.
I cannot resist the question.
Yeah.
How did you start as a CS PhD and got into sleep? Is it because you were sleep deprived, or how?
So it was actually the consciousness part that was interesting to me, and
I see.
I'll try not to grab.
So you consciously moved
Yes.
to, okay. Because that's something that I find interesting by itself.
Yeah, I found it interesting as well. And my hope was that the consciousness work that they were doing, they have like one of the leading theories of consciousness in the world, which I found very interesting. And I thought it seemed like there was potentially some usefulness to take that and apply it to AI, which I was very interested in exploring, but I needed to kind of talk with them a little more. So I've been excited by the opportunity to do that.
Yeah. Okay. It's wonderful. But it's sort of an interesting starting point.
Yeah, definitely. Which puts me kind of in an interesting position as not necessarily an expert in either of these domains, but here I am.
Yeah, I think this is extremely valuable to. We like non-experts because that exposes our weaknesses.
Right. So, yeah, we at the lab we realized this was very helpful. Realized that it could be useful for other projects as well. So that's how I kind of got assigned to this mouse brain data project, which I'm going to be talking to you about today. So just to get into the data, this is what our data looks like. This is actually 30 seconds of recordings. So the idea is we have these very high resolution neuropixels probes, which can each record about like hundreds of individual neuron spikes, which is a very, very detailed, super high resolution image of brain activity. I guess the highest resolution that you can currently get in vivo, but it's, as you can imagine, very large, very detailed, very processor intensive to actually work with. So we have about 80 terabytes total of data that we're trying to work with, which would be pretty much impossible to work with without CHTC resources.
Yeah, it reminds me, and I have to, once again, I cannot resist. We worked with a group that was doing similar with monkeys, and they said a monkey in 5-6 hours can generate work for me for 3 or 4 months. You know, that's what a monkey can do. I have to work a lot on this, and now you have mice, so yeah.
Yes. So this, each of these green dots that you're seeing is a single neuron spike. So this is only 30 seconds. You can imagine how much data it would be when we have 5 to 6 hours throughout the night. So yes, a lot of work for us to process. So getting to the aim of this project in general, it's somewhere between sleep and consciousness. They're sort of related because sleep provides us this area to study where you can study when a mouse is awake and conscious. You can study when it's asleep and unconscious, but also when it's asleep and conscious. So that provides a bunch of different modes to study to kind of determine the dissociation between function and consciousness, which is something the lab is interested in. So the animals are going to be asleep during the recording, and then different stimuli are going to be delivered to them, which is like an air puff or a flash of light or a sound. And then these recordings provide very high resolution mechanisms of what happens across different brain structures when they recover consciousness. So when they are woken up or when they remain disconnected during sleep. So what we're really interested in is how the neural signal is flowing during a state, which could be NREM, wake, or sleep. And so this is what we're ultimately going to get. This is a jittered corrected cross-correllogram. And so what this kind of models for us is which neurons are being driven and which neurons are driving the other neurons. So for us at the lab, this gives us a very high detail image of exactly what the structure is of the neurons in the brain and how it differs during the different stages. So in order to get one of these, we have as our input the spike trains that we kind of saw earlier, binned at 1 millisecond. And then we have this massive array of 2,800 neurons times the number of time bins. Remember, we have 5 to 6 hours of data times the trials, which is the sensory stimulus. So we're going to get this cross-correllogram for every single neuron pair. So that's 2,800 times 2,800 neurons. Quite a lot of interactions. And once we have this cross-correllogram, since we only care about the high frequency communication, we're going to jitter that by about 20 milliseconds. So it means shift it randomly. And we're going to subtract that from the original to generate this final jittered corrected cross-correllogram. And here is a bit bigger of a view of what that looks like. So the blue is driven by other neurons, and the red is neurons that are driving other neurons. So you can see this. I mean, I don't really fully understand it, but for the people that do, this is a very good look at what's going on in the brain. And I guess it's interesting for them that the REM looks much different from wake as compared to NREM. Like we would have expected wake and REM to look more similar, but this is very much still in progress. We're still working on it. So I'm not going to give you any results as to what that means, but it's interesting. Okay, so in case you didn't understand or didn't care about any of that, here's the meat of it with respect to what we're doing on CHTC. So as input, we're taking one file, which is one animal on one stage. So that's 2,800 neurons across 6 probes. And this can vary quite a bit in terms of the size of the data, which is something that, I don't know, we're kind of working on. The output then is going to be 36 jittered corrected cross-correllograms. That's one for each probe versus every other probe. So remember, we have 6 probes, so 36 total output. And we're doing that to kind of increase the parallelization of our processing. So without CHTC resources, this would take, you can see we did the math here, bare minimum, assuming it's taking 12 hours, which is kind of the lower limit, bare minimum, this would take 1,080 days to process. And the person who's working on this needs to graduate at some point, so that is not feasible. With CHTC, first of all, we were able to speed up the individual file processing a little bit as well by using parallelization within the file, which apparently you can do. And then theoretically, you would be able to run all of these files at the same time. Obviously, that's not how it actually works, but that can reduce our...
Can you break a file into segments? Do you have to do the file sequentially, or can you take segment? Because the file is, if I understand correctly, is time, right? You're measuring something over time. Can you cut it into smaller segments and then maybe glue it and therefore get more scale out?
It's possible. I think part of the reason that we're doing things this way is simply because that's how it was being done. So it was sort of easier to take what we were doing, so just put it on CHTC. But
Yeah, but if we want to make it, if it's important to make it less than 6 hours, or bring more capacity to the problem because you have many more files. Maybe one idea is to break the input and then stitch it together at the output. Because it doesn't seem to me that you really need to know what happened earlier to do some of the analysis downstream. I don't know, but I assume.
yeah, I think that makes sense. I'm not sure the extent to which we actually do need the whole night all at once or not. That's
So,
something I could ask.
and I would be happy to discuss it if you are, in my capacity as a computer science faculty.
Sure, yeah. So I'm going to get into a little bit of this ResearchDrive integration, which has been very helpful for us. So ResearchDrive is this secure shareable storage on UW campus's network. And so part of the reason that this was super helpful is because we have so much data, especially output files are very large. So it made a lot of sense to, instead of using the CHTC scratch space, which we got more of, so we have 300 gigabytes, but that's still pretty limited compared to what we actually need. It would be nice, we thought, to be able to just take our input data from another drive and put it directly onto a drive that we wanted. So this part right here is very fast between CHTC and ResearchDrive. That was fast already, but with the new ResearchDrive integration, we don't even have to worry about that at all. It just puts it directly on that drive. This is where all our data actually is. This part was kind of limiting for us. We found it was on a similar order of magnitude to actually transfer the output files because they were so big as to process them in the first place, which doesn't really help us. So that's why we wanted to use ResearchDrive. I guess I'll kind of skip past this getting started with CHTC. Sort of, we can ask about that if you guys are curious. But yeah, it was a little bit of a learning curve to get started with. I found the guide online pretty helpful, and mostly the people in office hours were extremely helpful to helping me kind of get familiar with this. Most difficult thing was setting up the Docker image, which I hadn't known how to do, but once I got it, it was fairly, didn't require much extra effort beyond that. So here's a look at what my submit file looks like. The ResearchDrive integration was fairly simple. We just sort of reached out to CHTC about this, had to get written approval from PI, and then that should have taken them about a day to set up, which it did ultimately. There was a bit of a issue where we tried to set up the wrong drive, but ultimately it took about a day. And then all I had to do was add a couple extra lines to my submit file. So this is the location of the ResearchDrive, and then transfer input files and output files are kind of the same as usual, but we just transfer them to this other location. So from my end, it was fairly seamless. But as I understand, this is a feature that is still in beta, so we did encounter some issues. But yeah, worked out pretty well. So for our current progress, like I said, this is very much still in progress. Once the current batch of jobs that we're working on is finished, that'll be 95 out of, I'm not actually sure how many, maybe like 360 jobs total. So very much we're still in the process of doing this. More jobs are being prepared. But our experience with CHTC has been great so far, and I'm sure that we will continue at the lab to use the resources here for other projects as well, in a similar vein to this one. So big thanks to the CHTC staff for being so helpful, helping me get familiar with this. And thank you all for listening.
Great talk. Nice talk. Do we have questions for Aydan? Well, I'll ask, you mentioned, ResearchDrive integration is in beta. What types of issues did you encounter in your?
So I guess one of the things that's a little like frustrating is normally when you're working on something like in computer science, if you have an issue, then it's like my issue, right? So I am responsible for figuring it out. So it was a little bit difficult to know when some issue happened, like at one point it was on the CHTC end, so we had to kind of communicate. So there was one issue with the file transfer not working, which kind of stopped us for a couple weeks.
Couple weeks, okay.
Yeah, I mean, so part of that I was like on vacation a little bit, but yeah, so that part ended up being slowing us down a little, but it seems like that's been resolved now, so we're good to go.
Okay, great. Do we have any other?